All right, I think we're ready to kick things off. First, I would like to welcome everyone to today's webinar. Just a quick introduction. My name is Yuan, and I am the content manager at Biocytogen Boston Corporation. I'll be your moderator for today's session. And I'm also joined by two amazing colleagues from the Biocytogen team as our panelists today, Dr. Leila Kokabi and Dr. Jade Xu. So let's take a moment to say hi and share a little bit, let them share a little bit about themselves. Leila, would you like to go first?
Hi, I'm Leila. Nice to meet you all. I will be responding if you guys have any question, especially related to in vitro. I'm a director of in vitro at Biocytogen, and I'm working more than 4 years here. So I'll be happy to answer any question.
Thanks, Leila. And Jade?
Hello, my name is Jae-Soo. I'm the Senior Business Development Manager at Biocytogen, responsible for the San Francisco Bay Area. And my research background is oncology with a focus on lung cancer drug development. And I was involved in advancing a drug into a phase 2 clinical trial. And I have been in a clinical facing roles for about 5 years, previously working as a field application scientist. Looking forward to today's talk and the panel discussion, and I'm happy to answer any question you may have.
Thank you both. Very glad to have you here today. So now I would like to introduce our speaker for today's webinar, Dr. Qiurong Wang. So Qiurong earned her PhD in molecular and cellular life sciences from the University of Wyoming, where she specialized in pharmacology and cardiovascular disease research using genetically modified mice. And following her PhD, she continued her work as a postdoc researcher at the Center for Fetal Programming, also at the University of Wyoming. And there she investigated the impact of maternal obesity on fetal heart development in sheep, as well as the transgenerational effects on cardiovascular function across offspring lifespans. Her outstanding research earned her a postdoc fellowship from the American Heart Association. And now here at Biocytogen, Qiurong serves as a regional business development director, where she applies her expertise in gene editing, drug-targeted humanized mice, and comprehensive pharmacology services, which all play a critical role in advancing drug discovery. So for today's webinar, Qiurong will be talking about a key area in therapeutic development, which is bispecific antibodies. And we will explore how bispecific antibodies are being used to tackle complex diseases with a focus on oncology and autoimmune disorders. And Qiurong will also introduce Biocytogen's in vivo animal models and preclinical capabilities, and walk us through case studies that show how these tools help accelerate the evaluation and development of bispecific antibodies. So before we jump into the presentation, just a quick reminder, the Q&A box is already open. So feel free to drop your questions in anytime during the session. We will leave some time at the end to answer as many questions as we can. All right, without further delay, I will hand it over to Dr. Qiurong Wang.
Thank you, Yuan, for the nice introduction. Hello, everyone. Today, I'm going to talk about the preclinical evaluation system for the bispecific antibody across different therapeutic areas. So today's talk, I'm going to first introduce some background information about the bispecific antibody, and then we'll introduce the animal models that we can use to evaluate the in vivo efficacy for the bispecific antibody, and then followed by the in vitro platform that we can help to analyze the mechanism of action for bispecific antibody, and followed by the summary and the future perspectives. First, let's talk about what is a bispecific antibody. So as you can see, this is the classic structure of the antibody. And this is the monoclonal antibody. Usually it has the identical two arms that identify the same epitope. And the bispecific antibody actually is engineered two different arms together to enable to recognize two different epitopes, either on the same or different antigens. As you can see that the bispecific antibody has been in the therapeutic area for a long time. It started the concept in 1960, and in the past 85 years, it has drawn a lot of attention in the research area, and actually it has made a lot of improvement in the last 10 to 15 years. And as you can see here in the landscape of the already proved drugs for the bispecific antibody in the world wide, and as you can see, since 2021, there's a lot of bispecific antibody has been approved in the market, and as lots of them are T-cell engagers. And this is the summary list for the bispecific antibodies that are in clinical trials for the autoimmune disease areas. As you can see that these list composed with lots of hot targets that are very important targets for the autoimmune diseases. Next, I'm going to introduce the animal models that can help to evaluate the bispecific antibody efficacies. So at Biocytogen, we have developed comprehensive animal models to support, to evaluate efficacy of the bispecific antibody, including the oncology models and autoimmune disease models. And also we have our human immune system reconstituted models to support either cancer or non-cancer models. And then I will go to each different model individually. First, I'm going to talk about the animal models to support the cancer bispecific antibody evaluation. So the first cancer model I would like to introduce is the syngeneic animal models. So the syngeneic model, that means the murine tumor cells is derived from the mouse and will be inoculated into the mouse background. So that is a syngeneic system. And usually people will think the syngeneic model are not ideal model to evaluate the antibody, especially the human therapeutic antibodies, because the mouse itself and also the tumor cells derived from the mice cannot recognize the human target. However, at Biocytogen, we have the specialty to enable this syngeneic system to help to evaluate the bispecific antibody. So I will divide the different systems, focus on the different mechanism of action of the bispecific antibody. So first, I would like to focus on the cell-bridging bispecific antibody. So as you can see here, we summarized the top 10 targets that has been draw a lot of attention for the antibody development. And as you can see that the CD3 is the top target, and followed by the PD-L1. And both targets has been synchronized in the clinical phase. and there are a lot of combination with CD3 and the other targets. So here I would like to take the bispecific antibody target HER2 and the CD3 as an example to show how we can set up a syngeneic model to detect this bispecific antibody efficacy. So this is the mechanism of how this bispecific antibody works. So this is the structure of the bispecific antibody. As you can see that the left hand has the HER2 binding arm, and the right hand side is a CD3 binding site. And as you know that CD3 is a surface marker of the T cell, and with the binding of one arm of the bispecific antibody, and the other arm will binding to the HER2 on the surface of the tumor cell. And once the binding of the CD3 binding site to the T cell CD3, and then it will activate the T cell and then cause the cytokine release, and cytokine will kill the tumor. So this is the basic mechanism of how this bispecific antibody works. And how can we build a a nice syngenic system to evaluate this bispecific system interaction is that first I would like to introduce our drug-targeted humanized mice. As you can see on the left-hand side is the normal mice, and it carries the complete mouse genome. And then our concept here for developed drug-targeted humanized mice is that we knocked out the mouse gene and replaced with the human gene. So with this replacement, the engineered mice will be able to express the human gene and will express the protein accordingly. So with the human expression of the protein, it will be able to give the drug target the binding site. So back to this system that I will talk about how we can use our humanized mice to set up this model. First, let's take a look at how we modify incorporate the human CD3 parts into the mice. So we employed this human CD3E, human HER2 double humanized mice into this system. So how we develop these mice, this is the targeting strategy. So we used our proprietary gene editing technology to first knock out the mouse extracellular domain of the CD3E gene, and then we replaced with the genomic region of the human CD3E sequence into this region. And then this will enable us to fully express the extracellular domain of the CD3E, and we still keep the intracellular and the transmembrane domain as the mice. And for the HER2 humanization, we first knocked out the mouse HER2 gene, and then we knocked in the full genomic region, not the full genomic region, but it's the extracellular domain of the genomic region of the human sequence into this genomic location. And then as the same design as the CD3E, that we still keep the intracellular and the transmembrane domain as the mice. And both humanized gene are driven by the mouse promoter. And after this modification, we validated whether this protein expression is correctly modified. So we have used the flow cytometry to validate the protein expression. As you can see that in the wild-type mice, we were able to detect the mouse CD3E, and in our double humanized mice, we were able to detect the human CD3E protein expression. And for the HER2 part, and we did the IHC staining, as you can see that the human HER2 are correctly expressed in different tissue type. And after we talked about the mouse part, and then we need next we will focus on the cell part, the tumor cell, because for the murine tumor cell, it doesn't have the human HER2 expression. It only has the mouse HER2. So now we introduced the humanized HER2 MC38 cells. So we did the genetic modification by insert the CDS sequence with the KIK promoter to ensure that the human HER2 is expressed in the murine MC38 cell. And after the genetic modification, we did the validation and show that the HER2 protein is correctly expressed in this human HER2 MC38 cell. So right now we have the mice and also have the cell ready, and then it can form a very nice syngenic system help to evaluate the bispecific CD3 HER2 antibody. So this is the case study that we implanted 500,000 of human HER2 MC38 cell subcutaneously into our human CD3E, human HER2 double humanized mice. And we give the mice 2 different dosage of the CD3 HER2 bispecific antibody. As you can see that higher dosage of the bispecific antibody showed a better tumor growth inhibition effect. Next, I would like to focus on the bridging receptors. So the antibodies that can bridge receptors from the same or different cells, and it can bridge the target either on the tumor cells or on the immune cells, or it can bring bridge between the tumor and the immune cells. So this is the list of the targets and the combination that focus on different mechanisms of action. So I will just take one example as a case study to show how this syngeneic model can be helped to evaluate this kind of bispecific antibody. So this case study is we use to evaluate the CD47 PD-L1 bispecific antibody. To evaluate the drug efficacy, we used the syngeneic model that we implanted the human PD-L1, human CD47 double humanized MC38 cells into our triple humanized PD-L1, human SERPA, and human CD47 mice to build this syngeneic model to compare the bispecific antibody versus their parental monoclonal antibody. As you can see that the vehicle group has reached human points about 14 days after the grouping started and on day 21, as we can see that the bispecific antibody showed a better tumor inhibition effect compared to the parental monoclonal antibody. Next is about the bridging cytokines and its receptors, or between two cytokines category. And this is the list of the humanized animal models that we can help to evaluate any bispecific antibody in this focus on the cytokine and its receptors. And these are the bridging cytokine and receptor mice, and these are the bridging cytokine mice. And I would like to take a very hot anti-tumor bispecific antibody, VEGF and PD-1 as an example, as a case study to show how syngeneic model can be set up to evaluate this bispecific antibody. So since, as I mentioned, that PD-1 and VEGF-A becomes a very hot therapeutic target. And right now there are two drug candidates at the clinical phase. One is from ASCO, it's imvonescimab, short by the AK112, and another one is from BioNTech, it's BNT327. And the first one, AK112, is at clinical phase 3, and BioNTech asset is at clinical stage 1-2. And here we give a case study show how we evaluate the efficacy for this particular PD-1 VEGF-A bispecific antibody. To build up this syngeneic model system, we inoculated the human VEGF-A MC38 cells subcutaneously to the triple knocking with human PD-1, human PD-L1, and human VEGF-A mice. And as you can see that the bispecific antibody showed a very nice tumor inhibition effect. So this is just a summary list of the animal models and the cell lines that will be useful for the cell engager bispecific antibody evaluation. And on the left-hand side, top is for the T-cell engagers. And we have developed lots of useful animal models and lots of combination of the target mice to help to evaluate either bispecific or multispecific antibodies. And also we developed the animal models for NK cell engagers. And on the right-hand side, as you can see that we have a lot of tumor-associated antigen humanized engineered cell lines. As you can see, most of the targets we have developed on the MC38 tumor cells, and we are keep expanding the human TAA on other cell line background as well, including EL4, B16F10, CT26, etc. And this is the list to categorize the products for the humanized animal models for bridging receptors. And this is the bridging receptor animal models for the tumor cells. And the middle section is for the bridging receptors on the immune cells. And the right panel is focused on the receptors on one on tumor and one on immune cells. And this is more comprehensive cell lines with the human TAA targets. And since we are heavily focused on the research and development, and we have released 10 to 20 new models on a monthly basis. So please contact us to have most up-to-date model catalog if you're interested in our models. Next, let's switch gears from the syngeneic model to the xenograft models. So the xenograft models has been widely accepted and used for the cancer research. And here at Biocytogen, we also support the xenograft animal models. And if the bispecific antibody itself is just targeted on the tumor itself, the xenograft model, just a simple one with the CDX or PDX on the with the immunodeficient mice, we have our own immunodeficient BNDG mice, it's equivalent to NSG. And those models will be sufficient to evaluate the bispecific efficacy. However, if the immune cells needs to come in play in the bispecific antibody efficacy, then the human immune system reconstitution models need to be used to evaluate the bispecific drug. So at Biocytogen, we have developed a huge database to help our clients to select the suitable target co-expression cancer models. And we have the RNA-seq data for all the CDX cell lines and the PDX models. And we can help you to find the perfect cell line and the PDX model for your desired expression targets. And also for each PDX samples, we have comprehensive patient data information. And also we have the RNA expression profile for all the CDX and the PDX models. And in addition, for the PDX model, we do have the database to further confirm the protein expression level. And we have the high throughput screening from the TMA and with the immunofluorescence staining to show the co-expression of the target protein expression. And also we have the IHC data to show the expression level. And with the choose the right CDX and the PDX model, here I just want to give a case study for using both CDX and PDX to evaluate the anti-EGFR MET antibody in the xenograft model. So as you can see here, we used our immunodeficient BNDG mice. And either with the CDX system inoculated with the NCI-H292 cell with 5 million subcutaneous inoculation onto the BNDG mice, or we chose the patient PDX sample BP0101 and with the piece of the equal size of the piece of the patient tumor samples implanted subcutaneously, and then we just gave the treatment of this bispecific antibody to see the drug efficacy effect. And as you can see that both CDX and the PDX showed a very nice efficacy data for this bispecific antibody. Next, just I want to showcase about how to employ the PDX model to evaluate the drug effect of the bispecific ADC. As you know that bispecific ADC is the payload linker with the bispecific antibody. And on the left-hand side, we use the PDX model, which is a non-small cell lung cancer tumor model, and to evaluate the bispecific ADC versus its parental monoclonal and ADCs, and it showed the bispecific ADC showed a better tumor inhibition effect. And on the right-hand side, it's just evaluate, it's the same PDX model, but we try to compare different dosage of the bispecific ADC and its benchmark ADCs. So the blue line indicates the lower dose, and the red line indicates the higher dose, and the yellow and purple line are both the HER2 benchmark ADC, and the green line is the TROP2 benchmark ADC. As you can see that the higher dose, both bispecific ADC showed a better tumor inhibition effect over the benchmark ADCs, and the higher dose of bispecific ADC showed even better inhibition effect. So as earlier I have mentioned that we can use our syngeneic model to evaluate the CD3 HER2 bispecific antibody. And here I just give you another example that how to use the immune reconstituted model to evaluate this bispecific antibody. So we can reconstitute the BNDG mice either with PBMC or with HSC. And here we use the PBMC reconstituted BNDG mice and inoculate with the CDX model to evaluate this bispecific antibody. And as you can see that with a higher dose of the bispecific antibody, it reached the tumor regression in the end of the study. And also with the PDX model, this higher dose is equivalent to the red line here. Also, it showed a very nice tumor regression with the PDX on top of the HSC immune reconstitution model. So the last example for the xenograft model is that for the leukemia CDX model. We just wanted to give you an example here. We used the human PBMC reconstituted BNDG mice inoculated with the Raji-Luke cell lines, and then we trying to see the bispecific antibody CD3, CD19, the drug efficacy effect. And we have the AVIS machine to monitor the progress of the tumor. And then as you can see that this is the blank vehicle group, on day 3, day 13, those mice has already been like a fully the tumor has been like a fully expanded in the whole body. And also for the PBMC reconstituted mice with vehicle treatment, it also is a little bit better than the vehicle, but it still had progressed a lot in this system. And this bispecific CD3 and CD19 showed a very nice tumor inhibition effect in this system. Lastly, for the model actually can be helpful for the different format of the antibody screening. So I would like to take example from a very hot and bispecific antibody from Amgen that targets CD3 and the DLL3 as an example. So as you can see here, this is the Amgen antibody. This is the drug formation, and actually here we just wanted to use different system to evaluate different drug formats. And with this two bispecific antibody, there are different formats of the bispecific antibody has been evaluated. And with the syngeneic model, we have inoculated the human DLL3 B16F10 cell line with our humanized CD3 EDG mice. And as you can see that compared to the benchmark Amgen analog, the format 3 actually performed better tumor inhibition effect. And on the other hand, we also employed the xenograft system, inoculate SHP77 onto the reconstituted PBMC reconstituted BNDG mice, and compared to the analog Amgen benchmark antibody, the format 3 also showed a better drug efficacy. So just this give you an idea that when you evaluate different formats of the bispecific antibody, both the syngeneic model and the xenograft model can be really helpful for the drug screening. So this is just a summary cell line list for the reconstitution models. And we have focused on different cancer types, and these are the cell lines that we have experience with. And if you wanted to have more detailed information, please feel free to contact us, and we will provide more information. Next, I'm going to talk about the bispecific antibody evaluation in the autoimmune disease and therapeutic area. So this is just a key therapeutic target summary for the autoimmune disease areas. So as you can see that those IL-4, IL-12, and TNF, lots of like hot targets in this autoimmune disease area. And again, this is just a recap what we have discussed earlier, that these are the bispecific antibodies in the clinical trials for an autoimmune disease. So at Biocytogen, we have developed the disease model platforms, and we support the inflammatory, autoimmune, and metabolic disease. So we can support different areas of interest, focus on the skin, respiratory system, digestive, joint injury, nervous systems, and the urinary system. All these systems, we can support the autoimmune and inflammatory disease models. And for the metabolic related disease models are listed here. So today I'm going just to give one quick example, focus on the respiratory system. It's using the OVA induced asthma model as an example to shed some light of how to use this disease model with the humanized drug-targeted humanized mice to evaluate the potential bispecific antibody. So here, I just wanted to mention that there are two potential therapeutic targets for asthma, and those are like existing drug in the market. One is the very famous dupilumab from Sanofi, which is target the IL-4R, and another one is target the TSLP from Amgen. It is the tezepelumab. And these two targets are like a very hot therapeutic drugs for the treating for the asthma disease models. And here, since we didn't have any chance to evaluate any bispecific antibody for the disease models yet, but here we just wanted to give you an example. We show using the combination therapy to use our humanized drug-targeted mice to facilitate the efficacy evaluation. So for this model, we use the quadruple humanized mice. We humanize the IL-4 receptor, and human TSLP and the human TSLP receptor in these mice. So the mice carry all these genes as a human gene in the whole system, and we induced the asthma by using the OVA. And then we give the treatment either by the monoclonal antibody and the combination therapy of the dupilumab and the tezepelumab. As you can see, the result showing here is from the cell count of the BAF, and these are the infiltration of the inflammatory cells, the eosinophils. As you can see, the combination therapy showed suppression of the infiltration of the eosinophils, and also we counted the eosinophils in CD45 positive cells, and it's even show a better inhibition effect. So that really like show that this quadruple humanized knocking mice can be a very nice model to help to evaluate the drug efficacy study in this area. So to continue that, for that same model, we also evaluated the total IgE in the system. As you can see that both combination therapy and the dupilumab showed a very nice inhibition effect for the total IgE released in the system. And also for the histology score, we also see that very nice inhibition effect from the combination therapy. So this is just an example to show you these mice can be very useful for the potential bispecific antibody targets, TSLP and the IL-4 receptor. And hopefully in the future, these mice can be useful for this kind of bispecific antibody evaluation. So next, I'm going to introduce our in vitro platforms to analyze the mechanism of action for the bispecific antibody. So I would like to take the Blincyto, which is CD3, CD19 bispecific antibody as an example to show you how our in vitro platforms can support you to evaluate the target's activation, fight, and the persist effect of evaluation of the in vitro for this bispecific antibody. So first, let's take a look at what we can do to support for the target-related bioassay evaluation for the bispecific antibody. So we mainly, actually for this part, is mainly trying to evaluate the binding effect of the bispecific antibody to the target cells. And here we can help to evaluate the target expression and the test article binding with cells and the ligand blocking effect. And the end result of these assays will be the flow cytometry. And next, for the T cell activation of the bispecific antibody, Blincyto, is that we use the system that we cultured the PBMC with different concentration of the Blincyto and trying to see different the T cell activation status. As you can see that with the concentration increase, the T cell activation is increased for both CD4 positive and CD8 positive cells. For the fight effect, actually, it's just looking at the T cell dependent cytotoxicity with this particular Blincyto bispecific antibody. Here we used two different systems to evaluate the cytotoxicity. So with the first one, we co-cultured the human PBMC with the DALI and also the different concentration of the Blincyto. And we used the LDH system to monitor the lysis percentage of the DALI cell. And as you can see that with the concentration increase, there are more and more DALI cells has been lysed. And another system we can help to evaluate these cytotoxicities to using the flow cytometry. As you can see here, we used the DALI cell with the T cells with the effective and the target cells ratio 1 to 10, and also we have monitored at 24-hour and 48-hour with different drug concentrations, and the cytotoxicity has been evaluated by the flow cytometry. And lastly, it's very important to see the T cell proliferation and the cytokine release in this system. So we just give a case study that we co-cultured the T cell with the DALI cell and gave the different concentration of the Blincyto, and to see how that increase of the drug concentration influence the cytokine release release. Here we monitored the human interferon gamma, and as you can see that with the drug concentration increase and at different time points, we saw the increased interferon gamma release to the system to indicate the persistent killing in this system. That is all about our in vivo and in vitro platforms to support the drug evaluation for bispecific antibody. And next, I'm going to give a summary and the future perspectives for this therapeutic area. So as we have talked before, that we have the leverage using our beautiful humanized drug targeted humanized mice, as we call biomice here, that we humanized the target gene, and those can be very useful to evaluate the target effect of the bispecific antibody. And also we have developed a comprehensive disease models platform, including the tumor models, syngeneic CDX, PDX models, and also the autoimmune disease models, as well as the metabolic disease models to help combining with our drug targeted humanized mice to evaluate different therapeutic application of the bispecific antibody. And in general, for preclinical pharmacology services, we can provide the in vivo, in vitro, from PK/PD to toxicity and the mechanism of action and analysis. And for the detection technology platforms, we widely cover the major detection platforms, including ELISA, MSD, flow cytometry, Luminex, IHC, and lots of fluorescence staining, and the different type of the cell assays as well. So hopefully with our biomice and our disease models and our pharmacology services, we will be able to help you to support your IND filing for your potential bispecific drug candidate. So for the future perspectives, here I would like to discuss with you some interesting, maybe can be a very hot area in the future for using the bispecific antibody. So there's lecanemab has been the first FDA approval drug for treat the Alzheimer's disease, and these give lots of drug developer the confidence to use the antibody to treat the neurological disorders. And here I would like to introduce a very beautiful targeting. There's a beautiful shuttling system. It's called the transferrin receptor mediated vehicle system. It can help to transport the therapeutic target across the blood-brain barrier. So there are two very hot targets in this area. One is the transferrin receptor called TFR, and another one is the CD98HC. So these are very useful the system to help to transport the therapeutic drug through the BBB, especially by using the antibody the binding can help to transport your therapeutic drug into the CNS. And here we developed a very nice animal model to evaluate the drug efficacy in this area, including our superstar humanized TFR1 mice, and it has been received a lot of attention and has been our number one seller since 2024, and also is a rising star for the human CD98HC mice. It's also another very beautiful model to help shuttle the test article through the BBB. And the next, I'm going to take this bispecific antibody as a showcase to show how to use our humanized TFR1 mice to evaluate the drug delivery, whether it can be a useful model to penetrate the BBB the barrier. So this is the bispecific antibody. It's a conjugate the heavy chain TFR1 antibody and also the anti-bcAe 1 antibody. So for this bispecific antibody, we detected the penetration of the bispecific antibody into the brain by evaluated by this IHC staining, as those red arrows show that bispecific antibody conjugate with the TFR1, it can really increase the neuron uptake. So that means that this bispecific antibody can really pass the BBB and entering the brain tissue. And on the right-hand side, it's the PK data showing that this using our humanized TFR1 mice to evaluate a different format of this bispecific antibody, and it did show that the concentration of this bispecific antibody can be detected, and it has the time-dependent manner, and both in the whole brain and also the functional area of the brain parenchyma. So this is conclude that the humanized TFR1 mice can be a very useful tool for you to evaluate this kind of a bispecific antibody with one arm of the TFR1, and you can conjugate the other arm with different targets, focus on the CNS disorders. So this is a very exciting slide, and when I see this, I just feel like it is like for antibody research area, it's really like blooming and trying to help to bridge all the therapeutic drugs together with the antibody. So antibody has a very important role to target to the specific cell or specific tissue. And with the conjugation with the antibody, either with nucleic acid or small molecule or protein peptide, or even more complicated system with different conjugation method, it will enable this antibody to a very wide range of the usage, and no matter what kind of format is going to conjugate with the antibody will be the key in this. I think will be in the key for the future therapeutic area, and to evaluate the target effect of the antibody. So our drug targeted humanized mice will be very useful in vivo animal models to help you to evaluate different conjugation antibody efficacy, and also combining with different disease models, including the oncology and autoimmune disease models, you will be able to see the drug effect. Hopefully this will give you some new ideas when you do the future drug development. So lastly, I would like to briefly talk about Biocytogen. So Biocytogen was founded back in 2008, and within the last 16 years of growth, and we have developed lots of different platforms, and also we have our two major sites. One is in Beijing, China, and one is in Boston, and both sites have the capability for the pharmacology services. And we also have the huge animal production center in China, which is #1 in Asia, and we have weekly shipment from our animal production center to the worldwide. And hopefully with our beautiful animal models and our pharmacology services that can help you to accelerate your bispecific antibody drug development. With that, I really thank you for your attention, and we are happy to take your questions.
Thank you, Chiurong, for the great talk. I think we already received many questions in the Q&A box, so let's take a look at those questions there. And to our audience, if you haven't submitted your questions yet, feel free to do so now. Okay, so let's take the first one here. What are the safety issues with T-cell engagers?
So I'll take this one. So again, like there is as we know, is a strong drug to use, but still always there is some issue. So major safety issue is cytokine release syndrome. So it can be caused by massive T cell activation and cytokine storm, and it can occur like within hours after administration. So what can be done to avoid that? We can it can be done step up dosing, gradually ramp immune activation. Or like use some intervention like cytokine blocker, such as IL-6 receptor blocker, or this can be also a little improved. Again, like this need to be done very carefully. And another issue which is also seen with some TCE, especially CD19 targeting bispecific and CAR-T, is a neurotoxicity, which also can be managed without use a corticosteroid to kind of manage the neurotoxicity. Another one is on-target, off-target toxicity, which, for example, in HER2 target TCE caused severe toxicity when HER2 was present in lung epithelium. So it's very important to kind of have a target, best target antigen selection, and choose tumor-restricted or highly overexpressed antigen. And again, like there is more, like, for example, T cell exhaustion, those kind of things, which kind of like, you're familiar, but those are three main one with the safety issue.
So thank you, Leila. And here's another question. Any good animal models to evaluate on-target off-tumor effect of bispecific antibodies?
I think I can take that one as well. So yeah, this is actually an excellent question, and it's very important when you want to do this kind of study. So so this is on-target, off-target is actually toxicity, actually very big challenge in this kind of, especially when you're doing the preclinical development. It's like, you can have like humanized mouse model, which can be like human PBMC or CD34 reconstruct mice, which we have a good example of in BNDG, or equivalent to NSG. So you're familiar probably with the NSG reconstruct. We have a kind of equivalent in the BNDG mice. So those can be used actually very good. It allows the bispecific to engage human T cell and human antigen, which is actually very good. And you can even detect the human cytokine storm through that. So another are the transgenic mice, which we have a humanized mice, such as like, human HER2. And also not only we do have a humanized mice, also we do have a humanized cell line, especially in the tumor. So for example, we have a Biocytogen humanized HER2, and then also HER2, Biocytogen HER2 MC38 that can be used together for those kind of study. And this can have a better control over antigen density and tissue expression, and good for antigen specific on-target, off-target toxicity. Again, there is other like non-human pyramid. We're not going through that. And also you can do some complementary ex vivo human tissue assay, but that was going to be a little like, it's still you need to do it in vivo. So I would suggest humanized mice. Yep.
Great. Thank you, Leila. Okay, next question here. How many mice do you inoculate to start with before you choose the usable mice for the project, since many mice will not develop same tumor size? And what is the cutoff tumor size do you use for the experiments?
Sure, I can answer this question. So usually we start with 8 to 10 mice with the overage of 20%, 20 to 25%, depends on the tumor model itself, because some tumor may have the very uniform growth rate. Some may have the heterogeneity of the growth, and then depends on the tumor performance, because we have extensive experience of different tumor models, and we will usually give you the suggestions what will be the overage part. And then, for example, if you the target the grouping treatment group for minimum 6 to 8 mice, we will start with 12 to 10 mice to begin with, and then to minimize any the different tumor size when we group into the treatment. And also, if you choose using our humanized mice, and the good advantage to use our pharmacology service is that we don't charge for the overage parts, and for both mice parts and also the procedure-related overage parts will be waived. Yeah,
included. Yeah.
Yeah.
Yep, I'll just add one more, two more points. So also, like, you know, that it very dependent. So for example, if we have a historical data and we know that we need 50% overage, we will do the 50%. But like for example, MC30 is something like more consistent tumor, 20-25% is enough. One more thing about when you choose the treat that, so you can either, like, very commonly 100 milliliter cube is the treatment start, but again, it depends on the tumor and then tumor model. So you want, and also it depends on your drug, the window for treatment, also like how fast grow. So it's multiple factor is involved, so we can kind of consult on that. Yeah.
Awesome. Thank you both. Okay, the next question here, how to choose between cGenic versus human immune reconstituted xenogenetic models?
I think I can take this question for you. So typically, a primary consideration when using the syngeneic model is the species match between the cancer cells and also the mouse model. And at Biocytogen, we offer not only the target humanized mouse cancer cells, but also the target humanized mice, which allows us to support your human antibody testing with both syngeneic models and the human immune reconstitution xenograft model. So speaking of the syngeneic model, because it provide a fully functional mouse immune system, so it will make it faster to set up the studies for you, and also more cost-effective options. And especially it's more suitable for the mechanism of action studies. And it also you can also have a better reproducibility due to the controlled immune background. However, the limitation of this syngeneic model is that because the mouse immune system is not as complexity as the human immune system, that's one part. And the other part is that the syngeneic model may not be suitable for biologics that require Fc binding to human immune cells. So on the other hand, if we are looking for a human immune reconstitution, xenograft models that can provide a fully human immune system. And then we can see the human tumor cells and human immune environment interactions. So this model is good for evaluation of your human Fc regions, human T cell, human B cells, and also some cytokine response. But the trade-off is that it includes a longer setup time and also a higher cost, and you need to manage the potential of the GVHD events as well.
Great, thank you, Jade. Next question here. Are there any concerns with sampling BNDG mice for drug exposure and PK? Are these mice sensitive to blood sampling over the course of the study?
Okay, I can take this one. So usually, like, you know, like the BNDG mice similar to NSG, so they are like more sensitive, you know, like they are more prone. So, and also, again, you don't want to compromise your study. So usually we have done a project both with BNDG mice itself or like reconstitute BNDG or PBMC, human PBMC engrafted one. So actually, like, because you also want to check the human PBMC engrafted. So weekly, once a week, to check, like, you know, to check the you know, for flow cytometry, it's okay, but which is also like 100 microliter per week, and that should be okay. That's fine. But if it's like, for example, PK analyzes, you know, kind of those kind of study, it's better to be a little, like, you know, be cautious, because you don't want to compromise the whole study. And kind of in those kind of study, we kind of suggest like you have like a rotating, you know, like for example, each group you have 6 mice, 3 mice this week, and then 3 mice in the other hour. So we can manage that way.
Yeah, like a staggered way. And also
Exactly.
we do have the technology for the micro sampling. So each time we can just sample 10 to 15 microliters for PK purposes.
Depends on the assay you want. Yep.
Mm-hmm.
Awesome. Thank you, Leila and Qiurong. So next question here. Can target humanized mice be used to evaluate the safety and toxicity of bispecific antibodies?
Yeah, I think I can take these questions. Recently, I think there are several scientific publications have suggested that if you are not using the non-human primates for your toxicity studies, maybe you can consider it genetic modified mouse or rat as your alternative models. And many of the pharmacology companies have also reached out to Biocytogen and use our humanized mice for toxicology studies to support their FDA and the applications. So I think we have about like 10 approved drugs for our clients for their FDA applications. And we also have some popular models for the bispecific antibody evaluation, including the humanized CD3E, CD28, and we also have humanized PD1, humanized 4-1BB mouse as well. And for other indication, we also have the humanized TSLPR mouse as well.
Great, thank you, Jade. I think we're running a bit short on time, so I will just take one more question.
You want me go? So I think there is two questions. I will just quickly answer. So why is it biospecific compared to ADC? I think was one of the first question. So like both are actually powerful treatment, a powerful drug. But again, like it's like multiple things can be involved in this kind of thing. Like why is advantage of BSC, for example? Again, each one has like their own limitation advantage, but we can see there are like different mechanism working, you know, for, and then for the bispecific immune activation, very strong T cell activation we see, which in ADC is not much. So, and also like for low antigen tumor, actually the bispecific is better than ADC. You know, the bsAb is a payload free, no need for the payload. And most importantly, it's mostly used in the solid and hematological tumor, specifically the one mentioned CD3 and CD19. There have been strong, good result on that, while ADC is mostly in the solid tumor. And of course, modular design is easier to tune in different formats for the bispecific. And also one more thing was about the why it needed a female, why we use female mice. So again, the people who work in the area and then actually do work in the in vivo, they know like male mice are more aggressive. So especially if you study the for the tumor and then you want to inoculate, and then you know you need to do the grouping based on the tumor, so you kind of recaging the mice. So literally, like, you know, you can see like they have prone to fight. So in this kind of case, if like very sex dependent, like for example prostate cancer, and we need to do on the male mice, so in this case there is no other option, so we'll just need to individually case them, which will be a little more cost effective and more space needed, but it can be done. But for the PK analysis, of course you can use a male mice if it's really more advantageous. And again, female mice have their own, for example, better immune response. So again, both can be worked, but like the main thing I think is like female mice is like much easier to work.
Great, thank you, Leila.
Yep.
Okay, one last question here. Can you elaborate a little bit more about the transferrin receptor mediated vehicle? What is the application of this system in bispecific antibody development?
So I will take this question. So as I explained in the webinar, that the transferrin receptor shuttling system actually is a very useful system to help to transport the therapeutic drug through the blood-brain barrier. And as we talked about the TFR, the transferrin receptor, it is a very unique factor that can both binding to the brain tissue and also go to the CNS. And also it has the effect to binding to the muscle itself. And we have expanded our validation data on the humanized TFR1 mice. And then why this is important is that you can actually bind the TFR1 on the one arm and engineer other like antibody targets on the other arm or the FC portion, and then to bring your drug, therapeutic drug into the CNS. system. So this will give a lot of like ideas for the bispecific antibody engineering part, so that can helpful to treat the neurological disorders by using the TFR to bring your therapeutic antibody into the CNS, into the brain to cure any neurological disorders that can be cured by the either antibody, or it can also conjugate either with antibody or conjugate with the antisense oligonucleotides. So there are very successful studies showing this system can be really helpful to help to cure the neurological disorders. So just to give you some idea, using this shuttling system to develop the bispecific antibody to cure this kind of disease.
Great, thanks for the detailed answer, Qiurong. Unfortunately, I think that's all the time we have for questions today. So a big thank you to Qiurong,
Yeah.
Leila, and Jade for sharing their insights, and to our audience for the great questions. And for the questions we didn't get to today, we will be sharing follow-up answers on Biocytogen's LinkedIn page. So keep an eye out for our post there. And if you missed any part of today's session, the webinar will be available on demand. You can request access or find the recording on Biocytogen's website or our YouTube channel. And if something comes to mind later, or if you would like more information about today's topic, our platforms or services, feel free to reach out through our website at biocytogen.com or follow us on LinkedIn. We will be happy to connect and continue the conversation. All right, I think
one thing. So I think if they
Yeah.
want to have the slide, I think they can request from you, and then
Yes.
we'll provide them. So, and if you want, like, we can have a, like, you know, like she can direct you to have a meeting with us, like, or like more specific need you have.
Yeah, you can find our contact info on the website or LinkedIn page. All right, I think that's all from us today. Thanks again for joining us, and we hope to see you at our next webinar.
Bye.
Thank
Thank
you.
you.
Bye.